For L9b, we'll discuss replication, a view at the molecular level, and we'll talk about DNA replication in prokaryotes, DNA replication in eukaryotes, and replication at the ends of chromosomes, the telomeres and telomerase. To begin with, one of the principles of all DNA polymerases is they polymerize from the 5' to the 3' direction. So this is set up as a double-stranded piece of DNA, two strands. So there's the 3' to 5' direction. Here's the 5' to 3' direction, and this 5' to 3', it has a free 3' OH, and the incoming deoxynucleotide, you have recognition between the A and the T, so this is your base pairing recognition, and the OH group attacks this phosphate, and we release pyrophosphate, and that gives us a 5' to 3' strand that's one nucleotide longer. If you notice, this was 2 nucleotides long, and now it's 3 nucleotides long. So things to remember about this particular figure: we elongate in the 5' to 3' direction. DNA polymerases can only add to an existing 3' OH group. This shows the active site of a DNA polymerase, and when we talk about transcription later with RNA polymerases, we're going to see that the active sites are essentially identical. So here's our 3' OH group. And notice it's attacking this phosphate. We have base pairing here. This is our template strand. We have base pairing here. Our deoxynucleotide triphosphate enters the active site, and we have 2 magnesiums that align and orientate the deoxynucleotide triphosphate through the negative charges on the triphosphate. So magnesium aligns this phosphate, this phosphate, this phosphate. And now it should be perfectly aligned with the template strand. And if the base pairing is correct, we will add a nucleotide to our growing strand. So this is N nucleotides on this strand. And now it's N+1 because now we have our phosphodiester bond linking what was our incoming deoxynucleotide triphosphate. So this is the DNA polymerase. This is the alignment of the incoming substrate, and we also have base pairing between the template strand and the incoming substrate. So this is our classic Watson-Crick base pairing. Here's our AT, and we discussed that in the preceding presentation. So here's AT, 2 hydrogen bonds, GC, 3 hydrogen bonds, and assume that the blue part that we're showing represents the geometry of the active site, and everything is fitting nicely. In fact, an AT has just about the same size from a molecular point of view as a GC, and this fits nicely into the active site. But we also have some other nucleotide interarrangements that will give us stable hydrogen bonding, but they don't fit well into the active site. So a G and an A can hydrogen bond through this carbonyl and this hydrogen of this nitrogen, and this ring nitrogen with this hydrogen, and the nitrogen of the A ring with its electron pair. So that's hydrogen bonding. But notice parts of the molecule, parts of the base paired structure is out of the area of the active site. So this is out of the area, this hydrogen's out of the area. Same with the G and an A. This part of the ring is out of the active site. A T and a G, this whole amino group is outside of the active site. So mismatches, these are called mismatch base pairing. These mismatched base pairings don't fit into the active site of the enzyme. So we favor ATs and we favor GCs. Comparison of the 3 DNA polymerases of E. coli. So we're going to have a polymerase 1, a polymerase 2, a polymerase 3. So polymerase 1 only has 1 subunit. Polymerase 2 has 7 subunits. Polymerase 3 has greater than 10 subunits. Now we're going to talk about exonuclease activity, and this has to do removing the nucleotide that we just added on the 3' end. We're going to see the importance of this when we talk about editing. So it has 3' to 5' exonuclease. That means it can start on the 3' end and remove nucleotides or deoxynucleotides, and in addition it has 5' to 3' exonuclease. So these are 2 exonuclease activities that some of the polymerases have and others don't. So 3' to 5' exonuclease, polymerase 1 has it, Polymerase 2 has it. Polymerase 3 has it. 5' to 3' exonuclease, only polymerase 1 has that particular exonuclease activity. 2 and 3 don't. Polymerization rate, polymerase 1 is pretty slow, 16 nucleotides per second. Polymerase 2, 40 nucleotides per second. Polymerase 3, 250 to 1,000 nucleotides per second. So this one's really, really fast. Processivity, that has to do with how far can it polymerize before it falls off the DNA. So anywhere from 3 to 200 nucleotides, this can elongate a chain before the enzyme dissociates. 1,500 chain elongation before the enzyme dissociates. And polymerase 3 greater than 500,000. So from these numbers, without knowing anything more, you could guess what is the main polymerizing DNA polymerase in E. coli. That is, which polymerase contributes the most to replicating the DNA, and you would guess it was 3. Mechanics of the replication fork in prokaryote. In order to replicate our double-stranded DNA, we have to separate the strands, and then we read each strand as a template to make a complementary strand of DNA. So here's our double-stranded DNA. We have an enzymatic activity that's called helicase, and helicase separates these strands. And upon separation of strands, we have DNA binding proteins that will bind to the newly separated DNA, and that keeps the strands from coming back together. So one of our strands, since these are anti-parallel strands, if this is the 3' end, and we're looking at the dark blue, that's the 5' end. And if this is the 5' end, over here, and this figure is blocking it, we have the 3' end. And remember, DNA polymerases, they polymerize 5' to 3'. So if this is the 5' end, and this is the 3' end, the complementary strand is 5' to 3'. On this particular DNA strand, if this is the 3', this is the 5', this is the 3' end, and we need to polymerize towards the opening replication fork, but we can't do that directly. We're going to have to do that indirectly by making short fragments of DNA that are actually being elongated away from the replication fork rather than towards the replication fork. This situation where we have one DNA strand that can be made continuously, that's called the leading strand, and it's 5' to 3'. And as this replication fork opens, the polymerase will extend the growing strand, and this is done by DNA polymerase 3. The other strand, if this is called the leading strand, the other strand is called the lagging strand. This was the first DNA we made. This was the second DNA we made. This was the third, and now we're making the fourth. And we have to start with an RNA primer. So we have an enzyme that's called primase that makes a short RNA primer. This is the 5' end on this end. This is the 3' end on this end. The DNA polymerase, which is going to be DNA polymerase 3, can extend this chain until it bumps into the next primer. Again, this is the 5' end of the primer. This is the 3' end. And now DNA polymerase 1, as soon as the polymerase 3 encounters the primer before, DNA polymerase 1 adds, and it removes the primer through its 5' to 3' exonuclease activity, and then it will start right here at this 3' end, and it will start at the 3' end, and it will continue the synthesis of the DNA strand until the two DNA strands are next to each other. So this strand has been extended all the way. This strand has been extended all the way. We don't have a phosphodiester length between this piece of DNA and this piece of DNA. So to recapitulate before I talk about this figure, we have a leading strand that's being made continuously, 5' to 3' by DNA polymerase. We have a lagging strand that's made discontinuously as the replication fork opens. And so we have an enzyme activity that's called primase that makes an RNA primer. that gives us a free 3' end. DNA polymerase adds to that 3' end, and when it hits this primer, DNA polymerase 3 dissociates, and DNA polymerase 1 associates with this region right here. It removes from the 5' to the 3' direction. It removes the primer, and then it will extend the gap that was created by the removal of the primer. And now we have an enzyme that's called DNA ligase, and DNA ligase ligates these DNA fragments together to make continuous chains. What I didn't mention is with primase, in order to make the continuous strand, we needed to make one primer for the continuous strand, whereas we need to make multiple primers for each DNA fragment that we make in the discontinuous strand. So the primase made a primer, and then we extended the leading strand. What this particular picture shows, it shows the symmetry at the replication fork. So this is a replication fork that we've expanded to show this figure. And it's actually moving from my right to my left. So it's opening up and it's moving from the right to the left. But there's another one in this bubble. This is called a replication bubble. Within this replication bubble, there's another replication fork. And because our directions get changed, the leading strand in the second replication fork is this strand, and the lagging strand is this strand. Because in one of these, in this fork, we're moving from the right to the left. In this fork, we're moving from the left to the right. So this becomes our 5' to 3' direction. This is our 5' to 3' direction. This is our opposite direction, which gives us the lagging strand. This is the opposite direction, which gives the lagging strand. So notice the symmetry between the two replication forks. So let's talk about ligase. So here was our RNA primer. Here's primer 1. This is the leading strand. Here's primer 1. This is the lagging strand. So the fork is opening away from the lagging strand, whereas the leading strand is being extended as the fork is opening, and the extension is towards the replication fork. So here's our first round of synthesis. So unwinding of parental strands and a second round of synthesis. So as this one opens up, our leading strand is being synthesized. And here's our second primer, and here's our second extension of DNA. And now we remove the primer, presumably with DNA polymerase 1, and then the DNA polymerase 1 extends this arrow to the end here. So here's our newly formed DNA between what are called Okazaki fragments. So these particular 1,000 nucleotide pieces of DNA that we make in prokaryotes. We've removed the primer, we've added more DNA, and now we need to covalently bind this arrow to this tail. And so that's called ligation of chains. So we fill the gap that we made by DNA polymerase 1, and then we're going to join the Okazaki fragments together by DNA ligase. So this is what ligase does. The 5' end has a phosphate. The 3' end has an OH group. And we just make a covalent bond between this phosphate and this OH group. But you could probably guess, we've done enough biochemistry right now, that that's energetically unfavorable. That has a large positive delta G. So we need to some way activate this complex so that we can make a covalent bond. And we're going to use either ATP or NAD. Some organisms use ATP, other organisms use NAD. So here's the enzyme, and it has a lysine, and this is the amino group of lysine. So the terminal amino group of lysine, with this electron pair, it attacks the phosphate group of either NAD or ATP, and it gives us the enzyme with the phosphate group that it attacked. It attacked this phosphate group and an adenosine. So here's a ribose. So this is adenosine. Here's a phosphate. And this is the enzyme. So we've covalently attached AMP to the enzyme. And here's our 3' OH group. Here's our phosphate on the 5' group. Here's our enzyme that has the AMP on it. This oxygen of this AMP enzyme complex that's negatively charged, it attacks this phosphate. And now it's going to be this this part, the AMP, is going to be covalently attached through a phosphate to this part of the DNA. So here we go. So the OH attacks O attacks here, and here it is covalently linked. Here's the phosphate, here's the O, here's the AMP, here was the phosphate that was on the 5' end of the DNA, and now this complex makes this phosphate much more positive, and the oxygen from the 3' end attacks that phosphate. AMP leaves, and now we've ligated together the two strands. So we've used ATP or NAD+. We made a covalent complex with the enzyme through an amino group of the lysine, so it's lysine, essentially AMP. The AMP becomes attached to the 5' phosphate, and then the 3' end of our DNA strand attacks the phosphate, and we make a covalent bond between our two DNA fragments, and we release AMP. If you take two rubber bands and twist them around each other, and then have one person hold the end of one rubber band, and what you're going to do is you're going to take the two ends of the rubber band and you're going to separate them, you're going to pull them apart, and what you notice is upon doing that, this segment becomes shorter. There becomes more, you actually get more twist and a shorter length, and the rubber band sort of kinks up, and this is actually supercoiling. This is an example of supercoiling. So as we're separating the replication forks, we're generating supercoils. This is actually positive supercoiling rather than negative supercoiling. Positive supercoiling can actually destroy the molecule. So we want to relieve this additional twisting that's happening at the level of the replication fork. So this figure shows something that's called the replicosome. How we can make the replicating apparatus move in the same direction while replicating both strands. So the replication fork we showed before, we had a leading strand where it was going 5' to 3', which we're showing in red. And as the replication fork opened up, it would give you chain elongation. It was basically waiting for the replication fork to open. And then we had another chain that was going away from the replication fork. But a problem with that is this huge protein complex actually moves together in the same direction. Half of it doesn't move away from the replication fork, and the other half moves towards the replication fork. In fact, the polymerase 3 is a dimer, and one of the subunits is replicating the leading strand, and the other subunit is replicating the lagging strand, and they're moving together as the replication fork opens. So we're going to talk about what's happening in the lagging strand that enables us to do this. So first, I talked about the rubber bands, and when you pull them apart, you make supercoiling that needs to be relieved at the replication fork, and we have an enzyme that's called topoisomerase, and topoisomerase is akin to helicases, and what it will do is as you generate the supercoiling, the topoisomerases relieve the supercoiling, and they covalently cut one of the DNA strands and wrap it around the other way. So it's actually taking the DNA, and every time it goes through a rotation, it cuts and reforms a strand, and that removes the supercoiling that's being generated. So like you clip the strand, you relieve the strain. You clip the strand, you relieve the strain. So you better clip them and twist them the other way and then reconnect them. These particular topoisomerases, they can move at 3,000 or 4,000 revolutions per minute. So let's look at some of the players. So here's our polymerase 3. There's a dimer. On both the leading strand and lagging strand, we have clamps. And these clamps hold polymerases onto the strands. Otherwise, they would fall off. So the clamps add to the processivity that we talked about. The processivity is the ability to synthesize long strands without polymerase dissociating from the strand. Well, the clamps add to the processivity. We have a helicase, and the helicase is separating the two strands. So there's the helicase. Attached to the helicase, There's primase, so as soon as we open this strand, we'll make our RNA primer. And notice, we don't show the RNA primer on the leading strand because it happened way over here. We only needed one primer, and then we did all of this chain extension. But for the lagging strand, we continuously need to make primers as we open up the FARC and we expose more lengths of DNA. So here's our primase. Here's our helicase that's taking the two strands apart in the replication FARC. And then here's our single-stranded DNA binding proteins. So polymerase 3 on the leading strand, that's easy. Here it is. It's moving from left to right. That's easy. But this one is also moving from left to right. How are we going to do that? And let's follow this strand. So what we've done is we've made a loop in the strand. So here would be a primer. So there's our primer, and the polymerase is moving from left to right. So it started polymerizing here. So we're adding DNA here, and notice we're moving towards the replication FARC. So this strand is being extended in this direction, and basically what this is doing, the polymerase is actually pulling this strand through its active site. So it's pulling the strand through the active site until this strand and this strand come together. And then this polymerase lets that strand go. It actually physically lets it go, and then it grabs this new strand in a loop, and now it will extend the DNA strand from this primer. So this primer would be over here, and now we're expanding the next DNA strand on the lagging strand. And then again, we have the ligase hooking these two DNA strands together. Polymerase 1 is going to remove this primer and extend this strand. Then it dissociates, and then ligase will ligate these two strands together, this and this together. So the first time you see this, it's a little bit complicated. Again, leading strand's continuous, lagging strand is discontinuous, but the two polymerase 3s are moving together, and they do this by looping. And when you loop this DNA around, you change its direction. So now when it's copying this particular strand of DNA, here it's moving away from the strand, but we put a loop in it, and now the loop allows us to move towards the replication FARC as one unit. So what I'm going to do is I'm going to post a couple of animations. One is a simple animation that shows how this is done, and the other one is almost a real-time animation that's absolutely spectacular. So I want you to look at the simple animation first, and then look at near real-time animation, and you get an idea of how this thing works. Properties of some eukaryotic DNA polymerases. So we've talked about prokaryotes, and eukaryotes are going to have a lot in common. And we're not going to generate a completely different mechanism just because you're a eukaryote or a prokaryote to replicate your DNA, but you could probably guess that the replicosome is going to be more complicated. So we're going to have, rather than 1, 2, and 3, we're going to have alpha, delta, and epsilon. So 3' to 5' exonuclease activity. Alpha doesn't have that activity. Delta has the activity. Epsilon has the activity. Associates with primase. Yes. So this is going to be the activity that's going to fill in gaps. Here's delta. It doesn't associate with primase. Here's epsilon. It doesn't associate with primase. Processivity, moderate, high, and high. So both of these are going to be involved with the majority of our DNA replication. High processivity requires pCNA. Remember this clamp that I told you about? In eukaryotes, this is called the proliferating cell nuclear antigen. So that's a protein you find in proliferating cells, i.e. dividing cells, and that shouldn't be surprising because we need that for our DNA replication. So alpha doesn't require pCNA. Delta and epsilon do require. A table I made of the eukaryotic DNA polymerases. Location, alpha's in the nucleus, delta's in the nucleus, epsilon's in the nucleus, beta, which is another DNA polymerase, is in the nucleus. Some of these are also involved in repair. So beta is involved in repair. And then gamma Gamma DNA polymerase is found in mitochondria. Remember, mitochondria have their own molecular biology. They have their own DNA synthesis, own transcription, own protein synthesis. Replication, yes, 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 no. Yes, but this is replication in mitochondria. Again, this is editing. Repair, no, yes, 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 no. So that's interesting. Mitochondrial DNA polymerases don't have the editing ability to clean up their DNA. Primase activity, yes. Delta, no, 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 no. 3' to 5' exonuclease activity. Alpha, no. Delta, yes. Epsilon, yes. Beta, no. Gamma, yes. So the replicosome is a huge complex that moves together. We saw that in our other figure, except that replicosome was in prokaryotes, and these are going to be the replicosomes in eukaryotes. So here's our leading strand template. Here's our newly synthesized strand. Remember, we put one primer out here, and then we've extended everything from there. And our leading strand is synthesized by DNA polymerase epsilon, and this is pCNA, this donut-looking thing that's holding epsilon on the leading strand. And then as we're opening up the replication fork, here's our topoisomerase that's relieving the supercoiling. Here's our DNA helicase and DNA polymerase alpha primase, so that makes our primer, and it makes the first few nucleotides of the DNA. Then it dissociates. Most of the lagging strand that isn't synthesized by polymerase alpha is synthesized by polymerase delta. So unlike prokaryotes, which use polymerase 3 for most of the lagging strand and most of the leading strand, we have a different polymerase for the leading strand and lagging strand. But remember, this is all one complex. So here's a picture that's very similar to the one we saw before. Here's our topoisomerase. And one difference is, on this one, the lagging strand is up on top and the leading strand is down on the bottom. So we've actually reversed the orientation. So here's lagging strand, primer, primer. Here's a lagging strand that we're now extending. Here's the leading strand. This was our first primer. Here's DNA polymerase epsilon. Here's the helicase opening up the replication fork. Here's the topoisomerase relieving the supercoiling as we're pulling the two strands apart. So notice, here's our alpha complex. Here's our delta complex. This is all, the primase is all in one complex. The delta and the alpha and the primase are in a complex with the epsilon. They're all moving from left to right. So here's the loop we've made, and now we're extending towards the preceding primer. When we reach the primer, the polymerase alpha will remove the primer and we will fill in the gap. Ligase will come in and we will ligate these strands together. So very similar picture to what we saw in prokaryote, but the name of some of our players have changed. So let's talk about replication in eukaryotes. And now we're looking at replication from a larger point of view. So now we will look macroscopically at replication, and we start our replication what is called an origin of replication. And in prokaryotes, the chromosomes in prokaryotes are circular, and you have one origin of replication, and the replication fork moves in both directions, and eventually they merge, and you now have two chromosomes. But eukaryotes have linear chromosome and they're not circular. And we're going to talk about the ends here in a minute, and we're going to also talk about the origins of replication. So let's look at one origin of replication in a linear chromosome. So here's a 5' end, 3', 3', 5' anti-parallel DNA. And we're going to have an RNA primer. So here's what's an RNA primer. This is a leading strand. Notice it's done continuously. Opposite that, remember our symmetry, we had another primer, another leading strand, and now these are multiple primers. That means this is the lagging strand coming from this fork, the leading strand going towards that fork. Here's the lagging strand, here's the leading strand. Eventually we ligate these strands together. So here's our 5' to 3', and the blue one is the newly synthesized one. So there's 5', 3', 5', 5 prime, 3 prime. And notice what happens is the 5 primed end doesn't complete its synthesis. And the reason was, in order to generate this strand, we went with a primer here at 5 primed and went over to 3 prime, and then we removed the primer, but we don't have any way of extending this DNA by our replication apparatus. We need an existing strand in order to extend the strand. Since our 5 primed end was RNA, and we removed that RNA, primase, polymerase alpha removed that RNA, that makes this DNA strand slightly shorter than this DNA strand. And the same thing in the opposite direction. This DNA strand is slightly shorter. So keep that in mind. So we have something of a problem since we need multiple origins of replication for 2 reasons. One, our chromosomes aren't circular, and each chromosome needs multiple origins of replication, because otherwise we have so many base pairs that if we only had one origin of replication per chromosome, it would take us forever to replicate it, but we end up replicating it in an hour or so. So we need multiple origins of replication for each chromosome. As a matter of fact, I've seen numbers as high as 20 to 30,000 origins of replication. So I'm not sure about the exact number, but it's thousands and thousands of origins of replication spread all through our 46 chromosomes. So notice, here's a replication fork, there's one, there's one, there's one, and eventually these come together and they merge. But the main thing about this, other than multiple origins replication, is the fact our newly synthesized chain is going to be shorter than our template strand. How do we get around that? We have, at the end of our chromosomes, we have repeating DNA sequences that are called telomeres, and we have an enzyme that's called telomerase that's able to extend the ends of our chromosome. So let's see how this is going to work. So here is the 3 primed end. Notice our 5 primed end is shorter, incomplete, newly synthesized, lagging strand. So here's our parental strand. And let's see if we see repeats. G, T, T, G, 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 T, T, G, 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 T, T, G. And this will be repeated many, many times at the end of the chromosome. And then telomerase has an RNA molecule as part of its active site. So this is the protein part of the molecule. This is the RNA part of the molecule. And that RNA is a complementary strand to the end of the chromosome. So telomerase, through its complementary RNA, can bind to the 3 primed end of the chromosome through base pairing. So here we see an AC binding to a TG, so TAG. In fact, you can move it over some more. GGG, CCC. This RNA can move all the way over to here. And then what we do is we use this RNA as a template to extend the 3 primed end. So the nucleotides you see in red, those use the sequence that's on the telomerase to extend the chain. And then once this chain gets long enough, we have enough room that the replication apparatus can put a primer in here and then extend our chain until we've completed the end of the chromosome. So we lengthen the end of the chromosome. That gives us enough space to create a primer going 5 prime to 3 prime, and then we can use our normal replicative DNA polymerases to extend that chain, and then we can use ligase to put the 2 chains together, etc. So this shows it in kind of detailed sequences. There's some things about this that are kind of hard to see because when they printed this, they got some of their base pairing off a little bit. So I put in another figure that doesn't have this level of detail. Here's our telomere repeats. AGGTT, AGGTT, AGGTT. Here's our newly synthesized lagging strand. We had to stop because we stripped out that primer. Here's our telomerase, and we use this RNA template to generate some more AGGGTT. So we extend this chain. And now we have enough room that we can make an RNA primer, and our polymerase completes the lagging strand. As cells get older, many cells lose their telomerase and their telomeres get shorter and shorter and shorter with each cell division. And that's one of the theories for aging, as you eventually reach a point. You've lost enough DNA off the ends of your chromosomes that you actually start interfering with coding sequences that actually put some sort of replicative limit on how many times cells can divide before they die or they become senescent. And that's one of the many theories of aging. So that completes L9b.